Okay, I decided to do this sequencing analysis to help your homework again because the record of the class is pretty huge and covers here and there. So I'm going to send this video instead to teach you stepwise how you pull out a sequence and how you do sequence alignment. Now I'm going to share the screen. So when you need to pull out a sequence, again, search for NCBI and you go to this website. You can select the database you want to use, like nucleic acid or protein, or you just type COVID-19. It will give you a lot of information. You perhaps can easily find the reference genome, which is the very first genome identified. You click this genome, you will find in this page so much information. So the requirement is to pull out the whole genome sequence. So but you roll down to the very end, you will say the full sequence starting from number 1 and 220K. This sequence, you are going to copy the full sequence because I ask you guys to do a full alignment. To copy this sequence to Word or any pad. So let's say this is Wuhan or reference sequence, you have to name it in this format. This format is critical. Then you copy the full sequences. I know it's huge, but you do not need to print, so I'm not worried about it. Let's go back to find some other sequences. As we said, there's so many mutants, maybe tens of thousands, have been deposited into the NCBI database. So how you find them? You can either search them or you can run BLAST. This run BLAST function actually allow you to find the closest sequence. So it automatically uses the sequence as reference, and if you do the BLAST, it will just pull out tons of sequence for you to review. So let it run. We are going to pull another sequence, okay? Okay, so here is the results, and it gives you 100 sequences, like from different resources. Of course, you are going to pick whatever you want. Again, don't pick those percentage of identity 100% because they're basically same. Choose some that has differences, more or less differences. Like how about we choose this one? When you click this, there's a sequence ID. So you click this ID and you can see this sequence is from another place. It was submitted 2020 January. And again, they give you a lot of information, but if you roll down to the end, it gives you the full sequence. So you will copy this sequence also. And paste to the same Word file, but make sure you use the right format, which you name it. Now you have 2 sequences, right? So the requirement is you have 5 of them using similar approach. So now you have 2. I'm not going to do 5, but they are very similar. So you copy all of this sequence and to align them. Remember, the alignment is called ClustalOmega. So you go to ClustalOmega, and here you choose DNA because we are comparing DNA sequence. And then you are going to paste both of the sequence, and then you submit. It will take a while to run, depends on your computer speed, but it will give you the results. Okay, meanwhile, I want to show you, while it is running, I want to show you how to pull out individual protein sequence. You still use this NCBI. I will pull out any sequence. Maybe we will use the reference sequence. You just go back multiple times. Okay, this is the reference sequence, and there's some ORF, means a coding frame, but they don't know what it codes for, but they're proteins they code for. There's very few of them, so if you go roll down, you will see there's some open reading frame here and there, and so those are the open reading frame, but there's a peptide it code for. So there's NSP2, which is a protein this virus codes for. There are, in the class I talked about a few, at least 5 proteins. They know the function already, right? Like proteinase like motif, they have structure proteins and transcriptase. So those proteins, when you roll down, for example, this is methyltransferase. This is the PP1A. So there's open reading frame. Let's say if we can find the spike protein. Strange, it doesn't find anything. So for example, spike protein, the gene is S, the gene product is spike glycoprotein, and CDS means coding sequence. So this DNA sequence code for spike protein. If you click CDS, it will highlight, this is pretty huge, and this spike protein sequence is used to design the full length of mRNA vaccine. So this actually is the mRNA vaccine sequence. Starting from ATG, which code for the methionine, the very first amino acid, it's always start from ATG, and end at TAA. This is stop codon. So from here to here is the sequence, DNA sequence. On the right side, it gives you the protein sequence. So you will pull out both DNA sequence and protein sequence and copy paste it to your homework. This is just one example. You need to pull out some other proteins like N protein, M protein. So those type of structure proteins. And in the class, I pull out 5 functional protein. You need to find them out and copy paste to your homework. So that is the second requirement. Let's go back to see our results. The results are already done. So This is only 2. And I ask you guys to do 5, don't forget. So you can see the Wuhan, the Yunnan strain, they are aligned. You can play with it, but I need you to copy paste the alignment for me. Okay, so second requirement is phylogenetic tree. You click the phylogenetic tree. In this case, there are only 2, so it's a very simple tree, but your homework will be a real tree. So you are going to give me the tree along with alignment, but your homework will be many, many pages, I guess. So I hope this satisfied the homework largely. The most difficult part, some of you guys asked me, how about the processing of S protein? I ask you to explore by yourself and write it down, whatever you find from internet. So those part, I'm sure you can find the answer on your own, by your own research. So the mutant, the England mutant, British mutant actually is coming from Italy. One of our students in the class talked about it, and you are going to find out the mutant from NCBI again, and also do the alignment. So this alignment, you do not need to align the full sequence. Instead, you will align only the S protein peptide sequence. Okay, so you basically pull out the sequence of S protein from the reference. You will pull out the sequence, this sequence of peptide sequence from that strain, England mutant, and do alignment, use ClustalW. So this time it's not a full genome sequence alignment. Instead, it's only S protein sequence alignment. Okay, so also use ClustalW, also send me the alignment results. That's all. You do not need to do tree this time as for the S protein alignment. So I guess this is what I want to share with you today. Please let me know if you have more questions.